diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
So all of this happens really quickly, all right? So it slows it down to take care of the steps, but it's still going to happen relatively quickly. And then the calcium or the calcium and calmodulin will have downstream effects, just like troponin and troponin C, have downstream effects in the muscle cell. All right, so now let's look at the third way.
When the ligand binds to the receptor site on the
So we look at the third fashion, or the third mechanism that is functioning. We have a combination of the two. So in the first one over there on the right-hand side of the board, we have the entrance of the binding of the receptor to its protein, causing activation of protein kinase A and subsequent phosphorylation. In the second pathway, we have the activation of the subunits opening a calcium channel. In the third pathway we're going to look at, both of those activities will occur. Okay? So it's even more complicated and it has a few more words to use. But it's really, if you understand these two pathways, that third pathway, even though it has new words and new molecules, is these two events occurring at the same time. So if you understand these components, it will work. May I erase this diagram? You guys have that one? Oh, L. So we still have a G protein that is activated in alpha subunit. Hormone binds to our receptor. The G protein interacts and is activated. The subunits separate. And the alpha subunit is activated by the replacement of GBP with GTP. Everything so far, remember I said these three mechanisms have the same first few steps. So here, in the previous one, alpha subunit interacts with adenylate cyclase. In this one, alpha subunit interacts directly with the calcium channel. Calcium channel replaces the enzyme and is the direct activity. Now we're going to have another membrane-bound enzyme. And this membrane-bound enzyme is identified as phospholipase C. It's not that much bigger, I'm just drawing it bigger so I can write the word to it. You haven't noticed already in biology, most of our enzymes end in ASE. All right, they don't always do, but if it ends in ASE, it's an enzyme. And this enzyme, in the diagram that you have in your lecture notes, looks like the molecule it interacts with comes from the cytoplasm, the phospholinositol. Right? It's not actually a cytoplasmic membrane. Phospholinositol is a membrane lipid. So this is not coming from the cytoplasm, but is found as part of the membrane. Remember I said we were going to be looking at the combination of these two activities? So we're going to look at the calcium formation first. One of the molecules released with phosphoinositol is broken down, is known as inositol triphosphate, or sometimes tris. So a heads up, a note well kind of notation. When you're doing lab exams, lecture exams, what do we wouldn't be allowed? Lecture exams or your homework where it asks you to name a molecule, you need to write out the full name. All right, you can put a key somewhere, a legend somewhere on the page, but on that homework for Tuesday, if you put in IP3 in one of those boxes, you'll only get half credit. All right, so you put IP3, but somewhere else on that page, you should say IP3 means inositol triphosphate. And again, on the fill-ins on the exam, I might ask you IP3 stands for what? So I want you to know those terms, okay? They are very long and complicated, and so it's, when you're practicing and everything like that, I understand writing IP3, but I do want you to know what they are. Inositol triphosphate opens calcium channels. All right, so this molecule is going to move into the cell. I'm getting to my blind spot here for you guys. And in the endoplasmic reticulum, such as the sarcoplasmic reticulum of a muscle cell, where calcium ions are stored, IP3 will open these calcium channels. And calcium will diffuse out into the cell. So in the previous example, calcium diffused into the cell from the extracellular fluid. In this case, calcium is still getting into the cytoplasm, but it's coming from inside the cell. All right? So IP3 is doing a function similar, not the same as, but similar to the calcium channel opening. And we have now more calcium inside the cell. And it will do additional cellular activities. The other molecule that is derived from phosphoinositol phosphate is diacylglycerol. And it stays in the membrane, unlike the diagram that you have in your lecture notes where it looks like it's diffusing in the cytoplasm. All right? So this is going to make this molecule known as diacylglycerol. Ever heard of triglycerides? So it might help you remember this is in the membrane by the fact that glycerides are lipid, and so this is a lipid-soluble molecule that's going to fit in that membrane. However, both of these are considered to be second messengers, even though diacylglycerol, unlike all of the rest of the second messengers we talked about, move around in the cytoplasm. So this is a second messenger. And this is a second messenger. Okay, which is unusual because it's not diffusing around in the cytoplasm. All right, so what does it do? It results in the activation of a protein kinase. Protein kinase C. What activated protein kinase A? In the first pathway we looked at? Cyclic AMP. And so, like cyclic AMP, DAG is going to activate a protein kinase, resulting in phosphorylation or occasionally dephosphorylation, only it's protein kinase C instead of A. So that's what I meant by combining those two different results into one G protein pathway. In a short video you'll see one of the effects of the calcium ions here is to join Yeah, the video I'm going to show you